In this video, we will discuss about chaperone mediated disease. This discussion is similar to our discussions that we have done during calnexin and calreticulum cycle. Only difference is that in this video we will discuss about a general idea how the problems in the chaperones will lead to disease, whereas in calnexin and calreticulum cycles we have focused only on the proteins that are affected or that requires calnexin and calreticulum in order to get their native structure that is essential for the further post-translation modification and for their native function. So let us start the discussion. The yellow region on the top of the figure represents the endoplasmic reticulum, whereas the green region represents the Golgi apparatus. So if there is a mutation in the proteins and that mutation leads to a change in native structure to extends to an extent that is beyond to an extent that is been uh, considered as a non-native structure by the molecular sense by the by the sensors that are present in endoplasmic reticulum that identify the structure of the protein, then they will direct them towards misfolding and misassembly, and this misfolding and misassembly will direct the proteins towards ERAD pathway, that is endoplasmic reticulum associated degradation, and they will take the proteins for proteasomal degradation. Whereas if the mutation is not enough to be detected by mutation is not enough uh, that is been detected by the sensor that has been detected by the sensor that is available in endoplasmic reticulum, then they will direct the mutated proteins towards further transportation into the Golgi, and they will then goes to the Golgi apparatus for further post-translation modification and proceed towards their final destination based on the uh, signal sequence or the oligomeric or the carbohydrate sequence that are present on the final structure of the proteins. And this mutations that leads to separation of the proteins into the misfolding and misassembly part, and then finally direct them towards the uh, ERAD mechanism, will leads to some of the disease will leads to disease, uh, and this disease can be taken care of by some of the external organic compounds or the small organic compounds of the acid which are been tested or which are being tested, and currently the clinic some of the compounds are being approved for clinical trial. So let us discuss some of these compounds. One of the compound is known as glycerol. What glycerol has been found is that it helps in facilitating protein folding and oligomeric assembly. That is, it helps the proteins to acquire the native structure and also helps in acquiring the native sugar sequence or the carbohydrate sequence afterwards. And this thing has been tested. Thing has been tested for the gene that is responsible for cystic fibrosis. So the cystic fibrosis, the CFTR gene, the CFTR knockout mutated gene, when it is being compared without glycerol and with glycerol, it has been observed that with glycerol the function of the CFTR has been improved. So this studies has been going on so that the glycerol or its modified form can be used for the cystic fibrosis. Another uh, compound, or we can say 4-phenyl butyric acid, that has been approved for clinical trial for urea cycle disorder because of its function that it helps in ammonia scavenger. That is, it helps to metabolize ammonia that are present in the uh, host host cells, so that can reduce the impact of this urea cycle disorder. And so this is just some of the examples that can compensate for the chaperones, or not, if not fully, but to some extent, such that the effect of the absence of chaperones can be reduced in a way that uh, the disorders that are generated because of the absence of chaperones can be reduced to a uh, threshold, can be reduced to a level that can be that can be used by, that can be tolerated by the human beings. So these studies are still under process. However, in future it is expected that from the initial success, that we will reach to a point where we can replace, we can find out compounds that can replace the function of chaperones. So that's all for this video. Thank you for your time.